Welcome to Multidimensional Models of Abnormal Behavior, Part 1, Genes and Behavior. Today we're going to go over some of the multidimensional models of abnormal behavior. In particular, we'll do a brief overview of genetic and neurobiological influences, familial and sociocultural influences, as well as biological and environmental factors that impact development and human behavior. But before we go too far, there are two important terms that I want to make sure that you know the definitions for. The first is etiology. Etiology refers to the cause or the source of a condition or behavior. So if we use an example of Professor B. Well has contracted influenza, we presume that contracting influenza, the etiology of that was coming into contact with the virus. The second term is multidimensional. This refers to a system of multiple influences, meaning that there are many different factors that will influence behavior or condition, and we can't assume that knowing one without knowing of the other possible influences will allow us to honestly understand the full story. Instead, we have to understand the many factors that influence an individual's experience and conditions. So using the same example of Professor B. Well, not only would we need to understand having come into contact with the virus, but we may need to know that this professor had a history of anemia, that they were experiencing extreme stress and exhaustion, and then in addition to that, they also came into contact with the virus. Those multiple systems of influences are the true reason for the outcome we witness. What do you think about when we say biological? For many people, when we think of biological, we think genetic. We think of genes. And we think that the strand of DNA might hold all of the secrets of existence for human behavior. But there are actually many biological systems that are influential in all of the conditions that we're going to study in this class. And all of these various biological systems are involved when we talk about biological perspectives. For example, central nervous system, autonomic system, the endocrine system, or hormones. All of these influence human behavior. Even when we talk about genes, most of the traits that we are examining are polygenetic, meaning they're influenced by many genes rather than there being just one gene that we can isolate. For example, if we think about the trait of being a genius, there is no single gene that's associated with genius. Rather, many different genes must come together in a precise moment and under particular conditions in order for the genius trait to manifest. And this is just one of the many reasons why such traits are fairly rare in the population, and why even when two parents with the same trait have a child, that child may not manifest that trait. Behavior genetics is one approach to examining the joint influences of genetic and environmental influences on behavior. Their goal is to examine patterns of traits within familial relationships in order to get estimates of how heritable that trait is. What do I mean by heritability? This is simply the proportion of variability in a trait or disorder within a given population that can be attributed to genetics. For example, the heritability of body weight is between 50 and 80%. So over half of the variability found in weight across all people in the population is directly due to their genes. Did you know that genetics are just as important to psychological conditions as they are to physical ones? Heritability for psychological disorders ranges from a low of 30% to a high of over 70%, with conditions such as schizophrenia and anorexia nervosa being among the most heritable of all conditions. And heritability for other conditions is also quite high, things like IQ and personality, although we haven't been able to identify the specific genes involved, alluding to the fact that they are likely polygenetic. There are several ways scientists can try to parse out the relative effects of environment and genetics. The first is a family study. In family studies, they examine the prevalence or the rate of occurrence of a trait or disorder in relatives of an affected individual and an unaffected individual. The goal is to determine the concordance rate between those with the disorder and those without the disorder. Just for clarity, concordance is the rate at which two people have the same disorder or do not have the same disorder. So if both have it, that equals concordance. If both do not have it, that also equals concordance. But if one has it and the other one does not have it, this is discordant. If there are familial influences, we would expect to see a higher concordance rate among individuals that are relatives versus non-relatives. But if you think about this, this can become pretty problematic pretty quickly. There's a lot of information that hasn't been explained. For example, it's not controlling for the mutual environment and the mutual genes. So we can't parcel out the environmental from the genetic influences. A second approach is an adoption study. Here we examine the prevalence of a trait or disorder in an adoptee, adoptive relatives, and biological relatives. If there is an environmental influence, we expect to see higher concordance rates among adoptive relatives compared to biological relatives. But if it is genetic, we expect to have higher rates in biological relatives compared to adoptive relatives. Can you imagine any potential problems of this approach? Maybe influences that have not been controlled for? For example, many individuals are adopted by relatives, or they're adopted within a few miles of their biological families. Most adoptions also occur within a similar social class or share the same ethnicity as the biological parents. So all of this means there may be environmental influences with some overlap between the biological and the adoptive family, but these are uncontrolled. A third approach is a twin study. Here they examine the prevalence or the rate of occurrence of a disorder in relatives of unaffected and affected people. In a natural environment, this would be comparing monozygotic twins to dizygotic twins. Monozygotic twins are what we would hear of as identical twins. They share 100% of genes. And dizygotic twins are fraternal twins. Here they share 50% of their genes, similar to any other sibling pair would be. If we see that monozygotic pairs are more likely to have a disorder than dizygotic twins, then we can deduce that there is a genetic component to this disorder. For example, if we find that monozygotic twins, or I'll say MZ twins, have 85% concordance, and dizygotic twins only have a 60% concordance, then there is a genetic effect. But I'll warn you, there still are likely to be environmental effects in addition to that. Can you think of any other problems with a study like this? One consideration is that people often treat twins as if they should act and behave similarly. And what you expect from people is often what you're going to get from people. These studies do not control for people fulfilling the expectations of others that they're going to be identical. It's also possible that twins are not really net representative of the general population. Their early biological experiences, things like sharing a uterus, often they will have shorter gestation. They have higher likelihoods of having to be separated from their mother and be in a NICU early in life, as well as their shared lived experiences as a twin, things like developmentally growing up at the same time, being treated as special and unique, also being treated as if they're not differentiable. This may make them a unique population. They're also experiencing all of their life events while at the same developmental stage as their twin. So this might actually increase their likelihood of having similarities, but once again, those similarities are not being controlled. So perhaps the ideal study would be looking at MZ twins separated at birth and adopted by vastly different types of people and comparing them to DZ twins with the exact same circumstances. And this would give us a better clue, perhaps a better solution, but that would be a pretty impossible study to design. One thing we also know is that genes don't act in isolation. Here we have what's called the diathesis-stress model. This is an important theory in the field, and it's going to show up over and over and over again throughout our class. What it proposes is that genes might actually be a diathesis. This is a condition that makes one more susceptible to developing a disorder at another point in time. So genes are the diathesis. They predispose us, make us more vulnerable to a condition down the road. And then environmental stressors may actually be the important factor that leads to developing that predisposition. So the environment acts as a stressor that triggers that genetic predisposition. As an example, risk for alcoholism among college students. Individuals that may have a predisposition toward alcoholism may not have discovered that this was triggered at all until they went to a college that was known for having a lot of binge drinking on campus. Interestingly enough, there's also evidence of a reciprocal gene environment model. This means that one can evoke the very environmental trigger that's needed for gene expression. In essence, genes aren't sitting around being lazy. They're actually pushing us into environments that could trigger their being evoked. And one great example of this would be children with a genetic predisposition for musical talent may go into a preschool environment and see lots and lots of toys, but find themselves drawn to the musical instruments. And as they have a chance to play with those musical instruments, it allows for the expression of their underlying genetic musical predisposition.